Hello, this is Group 10, and in this team video presentation for Biochem 424, we will be discussing the Hedgehog signaling pathway and its connection to cancer development. In this presentation, we want to establish what the Hedgehog signaling pathway is intended for in the cell and how dysregulation of specific points in the pathway can lead to mast cell replication. We will also touch upon current research being done in the field and address potential paths to take in the future for Hedgehog. The Hedgehog signaling pathway is highly conserved in the animal kingdom. As we see here, there are many iterations of the Hedgehog signaling protein. It is required for embryonic development and cell differentiation and is important for cell cycle activation throughout life. There's three types of cancers that can result from dysregulation of this pathway. Type 1 is ligand independent, type 2 being autocrine ligand dependent, and type 3 being paracrine ligand dependent.
In humans, the Hedgehog signaling pathway begins with any of the three Hedgehog proteins binding to PACHD1 and co-receptors GAS1, CDO, and BOC. Hedgehog proteins act as inhibitors of PACHD, which normally blocks the GPCR smooth from localizing in the primary cilium. As can be seen in the on-state figure, with PACHD inhibited, smooth localizes in the cilium, and its cytosolic tail is phosphorylated. This activates a signaling cascade, which results in the dissociation of Gli proteins from KIF7, a kinesin protein, and SUFU, a key regulatory protein in the Hedgehog pathway. Gli proteins are a family of transcription effectors. When the Hedgehog pathway is activated, Gli1 and 2 are processed into transcriptional activators via differential phosphorylation to induce expression of Hedgehog target proteins. However, as can be seen in the off-state figure, when Hedgehog is not bound to PACHD, these proteins are sequestered by KIF7 and SUFU, and Gli3 is processed into a transcriptional repressor via phosphorylation by pKa, CK1, and GSK3-beta. In simple terms, the active Hedgehog pathway results in the expression of cell cycle proteins, including cyclins D and E, as well as pathway inhibitors like PACHD1 as a form of negative feedback, whereas in the absence of Hedgehog signaling molecules, Gli3 acts as a repressor, thereby preventing the transcription of its target genes.
Of the 13 various forms of cancer that have dysregulated Hedgehog signaling, basal cell carcinoma and medulloblastoma fit into the type 1 cancer. These cancers are Hedgehog independent. These cancers caused by one of the two mutations. Usually, the PITCH1 receptor inhibits SMO, but when there is no signal, and activates SMO when there is a signal. But when PITCH1 is mutated so that it activates SMO without a signal, then it causes the cell cycle/differentiation genes to constantly be transcribed. Similar effects occur when SMO is mutated to be continuously on.
The Hedgehog signaling pathway is activated in around 95% of tumors. Currently, studies are being done on Hedgehog inactivators as anti-cancer agents. One example is sonigib, which inhibits SMO. In this trial, patients with advanced basal cell carcinoma were daily administered 200 micrograms of oral sonigib. Overall, most patients under this treatment had stable or improved disease-related symptoms. This study highlights the link between suppression of Hedgehog pathway and disease treatment for basal cell carcinoma. In summary, we have studied the Hedgehog signaling pathway, seen how it is linked to cancer, and how this knowledge may aid in the development of novel anti-cancer agents.